All right, good afternoon, everyone. Sorry for the delay. Today we have the pleasure of having a guest speaker with us, Dr. Yungsha Chen. She is a postdoc from Dr. Huang's lab. She finished her PhD just last semester, correct?
Correct.
So she's a fresh Dr. Chen. Her research specializes in 3D bioprinting, soft sensor fabrication, as well as composite polymer reinforcement. She's published in 10 peer-reviewed journals, very high-impact journals too. She's an experienced researcher. She's received awards for her research. She's received the Graduate Research Award, a Teaching Assistant Award, and a Certificate of Outstanding Merit in International Student Achievement from the University of Florida. Just another fun fact, she's also a co-author of mine on my NAMRIC paper. So
yes,
she's got
correct.
many publications, very well accomplished. So please pay attention. Also, I think Dr. Greenslet mentioned you might have one or two questions on your final exam from this lecture, so pay attention. Without further ado, the floor is yours, Dr. Chen.
Okay, thank you, Dan, for your nice words. I'm Yunxia, as Dan have introduced me. I just graduated last semester, so my undergraduate students started calling me Dr. Chen, and I asked them to stop because I haven't got used to that. So today, thanks to Dr. Greenslet, I have this opportunity to introduce my research to this class. It's kind of like very related because this is a non-traditional manufacturing class, and what I'm working on is 3D printing, which is very non-traditional. So let's get started. So the research topic is the fabrication of sensor-embedded heterogeneous brain simulants for the evaluation of impact-induced mild traumatic brain injury. Before we start, have anyone know traumatic brain injury before, or even experienced brain injury before? That's great, lucky you guys. So, however, TBI represents the traumatic brain injury. It's a major cause of death and disability. And every year, there are 2.5 million new cases of TBI just in the United States, and 50,000 people died per year as a result of TBI. However, as shown here, the majority causation of the TBI stays unknown. So it's really, why it stays unknown? Because the pathophysiological mechanism underlying brain damage is very complicated. So it can involve primary injury, which happens within a few milliseconds after the traumatic events. And also, it could involve the secondary injury, which could last for days or even weeks. Previously, people tried to use computational models, for example, the FEA models. However, its lack of physiological response and also needs experiments to validate it. So it's not a very good method. Another model people try to use is animal models. However, it involves a lot of ethical considerations, and also there are insufficient anatomical similarity between animals and human brains. So there are also limitations. So this motivated us to develop a research platform that can fulfill these gaps. So we would expect this model, which we call it a biomimetic brain simulant model, to be firstly have favorable biomechanical properties, and also have realistic anatomical geometry features with human brain, and also capable of integrating with intracranial sensors so that we can extract some deformation information inside of the brain out. And also we would expect it to be biocompatible with neural cells for future neurological studies. Research background and state of art. As we said, the human brain is very complicated, and one of the challenge to mimic human brain material is the material property of the human brain. So as we can see here, the brain is one of the softest tissue in human, and the elastic modulus, the stiffness is very, very, very small. So it's very soft material. However, previously people tried to mimic this material, and they failed to mimic the regional mechanical similarity of the brain. So challenges we have here, there are limited materials, and also there are strict requirements for this material to be processed. And also the material, the requirements for material stiffness tunable properties to match the brain regional mechanical properties are not there yet. And also there is some requirements for the materials biocompatibility with CNS cells. As we introduced previously, we would like this model to incorporate with some cell work in the future. Second aspect is about the fabrication method. So as shown here, the 3D structure is very complicated. It has like multi-layer inside of the brain. The most complicated area is the limbic system, which have like multi-layer, and they enclose the layers. It's very complicated to fabricate use traditional methods. So previously, people tried to develop some 2D brain models, like the 2D slices. However, it's lack of the 3D architecture and also lack of anatomical details compared with real brain. So the challenges here is fabrication of such kinds of 3D complicated architecture is very challenging. And also fabricate the heterogeneous brain regions, because different regions have different properties and they enclose each other. It's very complicated. And also fabricate of this nested structure resembled as the limbic system is very complicated. Another aspect is we would like to extract the deformation or other biomarkers disturbance inside of the brain out. So previously people tried to use different brain sensors. However, they are usually metal-based and very rigid. If we use a rigid sensor within the very soft brain material, what would happen? So there would be mechanical mismatch with this rigid material and the soft material, and there will be interfacial gaps between these two materials. And that can cause a lot of noises when we extract the signal out. So challenges here is it's difficult to avoid the additional damage caused by the rigid material implement. And also it's difficult to avoid these interfacial gaps, which can cause a lot of noise. And there are limited brain sensor materials that are ultra soft that can match the brain material stiffness. And also, as we said, there are primary injury, which happens at the millisecond level after the event. And also there are secondary injury, which can last for days or weeks. So there are multi-phase of the biomarker changes. And also at different regions, the brain would respond differently when we have impacts applied on the brain. And also, we would expect the sensor to be multifunctional, which means it can extract the temperature change if there are inflammation in the brain, and the pH change, because after the brain is injured, the pH would change inside of the brain. And also probably the strain change during the impact happening. So there are multi-phase, multi-regional, and multifunctional of the brain monitoring that is required. So challenges we have here is there are not many sensors that are capable to capture the acute response and the chronic response. And there are not many sensors materials that can measure the chronic biomarker change with long-term stability. And also there are lack of multi-modal sensors that can sense strain, pH, temperature, ion change, etc. at the same time. So summary for the challenges we have to fabricate a biomimetic brain simulant. There are brain-like region-specific materials needed. As for a lot of different manufacturing processes, the materials is is very important. And also the fabrication method for this kinds of nested structure is needed. And also the brain simulant monitoring sensor is also needed. So to address these three challenges we have, the overall objective of this research is firstly to fabricate biomimetic brain simulants with mechanical and structural similarity, and to fabricate brain simulants with embedded software sensors so that we can extract the interested signal out, and also to investigate the intracranial biomimetic dynamics for MTBI study using this biomimetic and sensor-embedded brain simulants. So, so far, any questions? Yes.
So, if you have sensors in the biomimetic brain thing, how would you like get swelling and pH and ion change from that? If like it's not really a brain.
Could you rephrase it again? Sorry.
So like, how accurate is the brain thing going to be to like have ion changes and swelling?
In swelling, you mean after the? Yes, I think that is a very good question. So usually when we measure a large disturbance of like temperature, pH, ion change, that should be very easy. It doesn't require the sensor to be very accurate because we have like enough room to allow the error happens. There are higher tolerance. However, in the brain, even if it is inflamed, the temperature could just increase from, let's say, 36.8 Celsius to maybe 40 Celsius, or even lower, like 39. So that actually have higher requirements for the sensor. It requires the sensor to be more sensitive, and also the ion changes. So I will introduce this later. We try to build a biconductive pathway, and this pathway can be selective to different ions. And with different ions, with increased concentration, the sensor will respond like sensitively. So this is something I will introduce later. Okay, thank you for your question. The overview for this research is, firstly, we'll try to fabricate a heterogeneous brain section. And we demonstrate this part with printing the limbic system. And secondly, we would like to develop the sensor array and to measure the disturbance change inside of the brain. So this is very important here. We try to achieve this method. We try to firstly develop the material system. And this material system can be used for the brain cortex fabrication, which is in larger volume, and people consider it as homogeneous. And also, we try to develop stiffness, tunable gelatin microsphere for the brain limbic system, because the stiffness is tunable so that we can match the stiffness of different regions of the limbic system. And also we try to develop a conductive gelatin microsphere, and this will be used for the fabrication of brain sensors. This is the first section, the material system. And then for the second section, which I read it in chapter 3, is about the manufacturing approach. Here we developed a nested printing process. It is developed based on the embedded printing, the regular embedded printing. But in this way, in this nested embedded printing method, we are able to fabricate multi-layer enclosing the structure. And this will use yield stress fluid, which we will talk about later. And the third task is about the sensing system. So we have the sensor that is implemented, embedded of the cortex, and it can, and we can implement them into different regions of the brain. So when the impact is applied, different regions will respond differently, and then we can use the sensor to acquire the the information out. So this is an overview of this research. And then I break this down. For each task, I will break it down into subtasks, and then I will go to the details in each chapter. So for the first task, the material system development. So remember, we have 3 different objects. The first is to create the homogeneous large volume brain cortex area. And the second is to develop a stiffness tunable Judson macrospheres so that we can match the regional stiffness of different regions of the brain in limbic system. And also we developed the conductive Judson macrosphere, and this can be used for brain sensors. So firstly, why Judson is used for the brain simulant fabrication? It's biocompatible and it's printable so that we can use it with 3D printing, and it's tunable. So we can tune the mechanical property to match different stiffness of different regions of the brain. Firstly, the brain cortex. So the brain cortex is like in large volume, and people consider it as homogeneous. So in this way, we just use the Judson macro polygons with mechanical fragmentation. And with different fragment time, the Judson macro particles can be broken down into different sizes. Here is statistical results with increased mechanical fragmentation time. This can decrease from 285 to 45 micrometer. So this will be small enough for us to print. Secondly is the brain limbic system, Judson macrospheres. So we control, so there are different parameters that can be controlled in this process. We can control the composition of this composites, the pH modulation during the Judson formation, the liquid height, which will apply different shear force, and also the vortex speed. And this will help us to get different particle size. And here is statistical results with different gelatin and gamma arabic ratio with different pH value in the gel formation environment, and with different liquid height when we put it into the shaking bed, and also with different vortex speed. The particle equivalent diameter can be manipulated and get the size that we like. So after we get different morphology or different size of the particles, we can tune the mechanical property of the composites. So to better understand why this could work, we can have a closer look at the composites we used here. So firstly is the Judson solution, which we dissolve the Judson powder directly. And secondly is the Judson macrogel, which can be fabricated using either the mechanical fragmentation or the complex conservation process. So this is the composite we are going to use for the brain fabrication. And the macrogel can be used as the macropolygon, which is the mechanical fragmentation macrosphere from the complex conservation or the conductive macrosphere. So firstly, we can adjust the constitutional particle size with either the fragmentation time or the different parameters we controlled during the gel formation process. Or we can adjust the packing ratio between the Judson macrosphere and the Judson solution. So using these 2 ways, we are able to control the mechanical property of the final composites. And here is the statistical results with like decreased particle equivalent diameter. The Young's modulus of the composites will be decreased as well. And also with increased dehydrated macrospheres, the Young's modulus can be also decreased. So in these 2 ways, we are able to control the stiffness of the final material. Here is a research showing how different regions of the brain, and their corresponding Young's modulus. So we can see there are large variations. And this is spectrum of the stiffness of the materials we developed. So we are able to achieve the Young's modulus from 380 to 4,300, and this will like cover the stiffness of different regions of the brain. For example, the corpus callosum and the brain cortex. And we also did the like in addition to the Young's modulus, we also did some rheology property testing, and also the Bodies line is the samples we measured, and it can cover the reported data showing in previous literatures. So the conclusion is the achievable tunable range of the viscoelastic property of the brain-like material covers the typical range reported in previous literatures. So in this section, we covered, firstly, the tunable morphology of the macrogels, which we use the complex conservation by tuning the different parameters during the gel formation process, and also the mechanical fragmentation. And the resulting mechanical property of the composites have the range of the have enough range to cover the mechanical property reported in previous literatures. So the second task we are going to do here is to develop the nested printing for brain limbic structure fabrication. So before we start, I would like to introduce the embedded extrusion printing to you. So we all know the extrusion printing, requires a supporting base, and it requires the material to be yield stress material, which means before you apply any shear on it, it will behave like solid-like. But when you apply a shear force that is large enough to exceed the yield point, it will behave like liquid-like. So a very typical yield stress material would be the hand sanitizer. You will see the air bubble trapped inside of it, and it will trap there forever. Until you shake it, it will be liquid-like, and the air will be released. So here we use the yield stress material in this printing, and we use nested printing. Nested printing is advanced form of embedded printing, which belongs to extrusion printing, and which is a method of 3D printing. To compare the difference of the embedded printing, which is a traditional way, and nested printing, which is an advanced way of embedded printing. So the similarity of these two methods is both of them will need a support base and require it to be yield stress. And the difference is the nested printing will deposit the structures inside of the pre-existing structure, pre-deposited structure, so it can form multi-layers if we keep depositing the material into the pre-deposited structure. So this is our setup for the brain simulant printing. And as we mentioned previously, we will use this gelatin composites, which consists of the gelatin microgel and gelatin solution. And this will behave like a yield stress material. So when the shear stress applied is less than the yielding point, it will behave like a solid-like material. And when the shear stress we applied on it is larger than the yielding point, it will behave like a liquid material. So this will allow us to deposit the material on site, and after the nozzle travels away from the region, the shear force disappeared, and it will trap the deposited filaments inside of it, because it will recover to a solid-like material. This is, however, we would like to, just like a lot of other printing process, nested printing also needs the improvements of the depositing fidelity. So a very common phenomenon happens here is upflow. So upflow means when we try to deposit the material at the target position, the filaments will actually flow up a little bit. So we would like this material to be deposited right here, but because of the hydrodynamics inside of the around the nozzle area, the filaments will actually flow up a little bit. And also, we would like to deposit the material and expect it to be perfectly spherical. However, it will be like a droplet, also due to the hydrodynamics around the nozzle region. And another kind of flow is the interfacial upflow, which means when we deposit the materials inside of the pre-existing material, the enclosing structure, also called the previous deposited structure, will be distorted because this region will have like shear force applied on it, so it will become liquid-like, and also the hydrodynamics will help the previous deposited structure to be distorted because it behaves like liquid-like and it can flow around. So in this section, we would like to mitigate this upflow phenomenon inside of this nested printing. So we identified different, so this is the printing result in our experiment that we observed. We figured that by controlling the printing speeds, the nozzle diameter, and relative viscosity between the depositing ink and also the matrix, we are able to mitigate the filament displacement. And similarly, the filament distortion can also be mitigated by optimize the printing speeds, the flow rates, and also the relative viscosity. As for the interfacial upflow, here are also some parameters we can use to tune it and to achieve the optimal results. So other than the defects or the printing fidelity loss during the depositing process, there are another factors that could affect the final printing fidelity. We call it the volume deposition induced printing fidelity loss. So that means when we try to deposit a multi-layer structure, the outer layer of the structure will be distorted. And here is the 3D reconstruction of the printed structure. And this is the first enclosing layer, like the outer cube shown here. And this is the secondary enclosing layer, which is a spherical. That means when we deposit the second layer, the first layer will be distorted. And when we try to deposit the third layer, both the second layer and the first layer will be deposited. So to mitigate this kind of deformation, we used the pre-compensation. Instead of depositing a perfect cube, we pre-compensate this based on the volume that can extrude out. And then without pre-deposition, we see the printed cube and the sphere are all deformed. And with the pre-compensation, we can get decent enough structures. And that's it. This is the process. We try to mitigate the filament displacement, filament distortion, and also the pre-compensation to mitigate both the printing upflow induced shear fidelity loss and also the deposition volume induced shear fidelity loss. And then we applied this method to our brain simulant printing. We have casted, we have like different, so the first depositing structure, second depositing structure, and third depositing structure. And we have printing result here. Without the mitigation, it can have huge upflow. And also, and with mitigation, we have like clear boundaries and also well-defined regions. So this is our brain simulant printing results. So how to evaluate the brain regions if we print it like in 3D? And also it's very difficult to use the CT scan to get the printing results because the density are very similar. To visualize the printed regions, the 3D regions, we use the 3D structural similarity comparison by slicing them into 2D slicings and then use image processing and the segmentation. We are able to like identify the printed region and then stack them together to achieve the 3D reconstruction and compare the 3D reconstructed region with the design structure to get the printing area. And after the construction, we see the printing result is very accurate with mean error within 2 millimeters. So this section, we developed a mitigation strategy and printed the brain simulant with high shape fidelity. The next task is to fabricate the sensors inside of this brain simulant. We used the material called MXene, which is a very conductive 2D material. And the MXene is usually prone to be oxidized, especially when worked in the liquid environment, such as the brain. So we applied the anti-oxidation process by using the gelatin to encapsulate at the surface. So this can prevent the oxidation of the material. And this material can still be conductive. Here is some measurements. Compared with the pure gelatin, the MXene gelatin have huge improvements regarding the conductivity. It has around 600% improvements of the conductivity. And we have sensors printed inside of the gelatin. So time is almost up, so I'm going to hurry up here. And also, as we mentioned earlier, the rigid material, the rigid sensor will have the interfacial gap because there are interfacial mechanical mismatch. So here we have the gelatin MXene material, which is very soft and similar to the brain material. So there will be no interfacial gap, as shown in the SEM image here. Here is a response to the strain and strain rates. And the sensor response to pH, sensor response to ion concentrations. In the last section, we are going to incorporate the material system, the fabrication technology, and also the sensor into the sensor-embedded brain simulant. We have implemented the sensor into the frontal lobe, the occipital lobe, and then and also with centralized sensor distribution in this way. And also we applied some impact on it and acquired this signal out. Here is the deformation of the brain under the high-speed camera. This camera can acquire the images with 1,000 frames per second. And we can see the propagation of the deformation inside of this interested area. And the sensor can get about 60% resistance change. And also we try to use the centralized sensor configuration. So when we apply the impact from different directions, the sensor at different direction. For example, the B1, B2 and B1, they will respond differently. And the distributed sensor, we apply them into frontal lobe and occipital lobe, and we also observe the different sensor deformation. And different protection gears, we also observe the different response. So I think that's the overall of this research. So we have 2 more minutes. I can take one question. If not, okay. If not,
Dr. Bruza mentioned, did you prepare a quiz for them?
Yes, but so there will be, I prepared 2 quizzes. Dr. Greenslice will pick which quiz, and she will put one of the questions into your final exam. So get prepared.
I thought we were going to go over it in class to help them study a little.
No, not in class, but in the final exam. So get prepared, guys. So thanks to all my collaborators and PI. And also, some of you guys, we have 2 more minutes, so some of you guys probably have attended to Dr. Yong Huang's class before. And here is what our lab is working with. If any of you are interested in participating in some lab research, feel free to reach out to Dr. Huang or me. So we have traditional manufacturing process and also 3D printing, and also the design and fabrication of microphysiology and engineered living systems. So if anyone is interested, please let us know. Actually, this work is helped with a lot of my undergraduate researchers, and I think that will be a great opportunity for you guys if you are interested. Okay, I think that's all.
Any questions? All right. I guess, yeah, we're right at 11:30, so if nobody has any questions, we can adjourn. Thank you, Dr. Chen, for stopping by.
Okay, thank you guys.
So we'll wrap this up now. And once again, last call. If anybody has not picked up an exam, yeah, I'll give you your exam today. Anyways, good luck on your makeup exam. And Dr. Greenslice will be back on Friday.